how do scientists study the DNA sequence of a gene? Hui-Ren, one of the technicians in Dr. Shih's lab, is sequencing the gene for MAO A by using radiolabeled DNA. She loads the DNA sample she is studying the sequence of into the gel. Here, she is loading 4 lanes, each containing one DNA base, A, T, C, or G, along with a blue dye to show how far the solvent has traveled. The technique of electrophoresis that we see here will pull apart the base pairs of DNA according to size. Electrophoresis is the process by which molecules, such as proteins or DNA fragments, can be separated according to size and electrical charge through the application of an electric current. The current forces the molecules through pores in a thin layer of gel. The gel can be made so that its pores are just the right dimensions for separating molecules within a specific range of sizes and shapes. Smaller fragments usually travel further than large ones. This process is also called gel electrophoresis. When the electrophoresis is done, the plate is removed and washed. The plates are separated in order to expose the gel containing the radiolabeled DNA directly to a filter paper. An X-ray sensitive film will be overlaid onto the gel and then exposed overnight. Here, our researcher is able to see a picture of the order of bases in the DNA sequence. Over the past few decades, researchers have modified and automated these techniques to determine the entire human genome, which enabled the Human Genome Project to be completed in 2003. This was a major milestone in life science research. Hui-Ren is now able to read the developed film from the gel so that she can determine the base pair sequence of the DNA. She is writing down the sequence for the MAO A gene.
That's okay. That's okay. It's done.